All right. Hi, everybody. I'm Josh. I'm talking today about rat, mouse, and stoat survival of past 1080 drops. For the last year or 10, I've been crunching numbers on this subject, numbers generated by the footprint tracking tunnels that we've heard a lot about today. And I'm joining the growing school of thought that background food really matters. My data set supports the notion that background food levels have an important influence on survival rates of rats and mice through 1080 drops, and therefore probably of other control methods too. I suspect the same goes for stoats and feral cats, but I haven't actually been crunching numbers on this. I've just been doing some preliminary eyeballing of the somewhat patchy stoat tracking data set. That background food should matter is not a new idea. You might say it's not terribly surprising, but as Graham said, we in DOC have done lots of 1080 drops right when background food levels are highest in the middle of mast years. So I've estimated rat survival rates for about 100 1080 drops. Here they're sorted by the rat survival rate. Each slot on the x-axis, or each blue bar, is a different 1080 drop, with the worst kills on the right-hand side, and those on the left without a blue bar are the ones with 0 rat footprints on the post-1080 survey. And if 3% was of survival of rats was our target, which is a 97% kill, then we've missed it about a quarter of the time. For mice, it's worse. We've only gotten less than 3% survival of mice at about a quarter of the 1080 drops. More than half the time, a third of the mice survive, and about a fifth of the time, no mice were poisoned at all. In fact, quite often the mouse tracking rates have gone up after a 1080 drop. Sadly, I can't show you a chart like this for stoats, because surveying for stoats isn't as simple as surveying for rodents, so we don't monitor stoats sufficiently well to produce such a chart. My number crunching of rodent tracking data seeks the commonalities between successful versus less successful operations by modelling variables such as background food, bait sowing rates, competitor density, etc., whilst controlling for bad luck with the weather and other random variables. In the course of this number crunching, I've had to step back and think about how poisoning works. For a rat or a mouse to die in a 1080 drop, it must first encounter a pellet, and then it must eat a meal of this pellet. And its first meal must be a proper meal, 1080 being an acute poison with rapid onset of toxicosis. So what creates and ensures an encounter, and what leads to acceptance of a meal? The way I see it, the probability of a bait encounter, the combined result of bait spread, that's how many pellets and how evenly they're sown, with bait competition, that's other animals eating the bait before the target animal can get to it, field life, like baits becoming non-toxic in the wet, and the ranging behaviour of the target animal, like how far and wide and how fast the animal's moving and for how much of the time, which probably changes, I reckon, in relation to things like density, as Dean discussed, but also in relation to background food, like home ranges possibly shrinking when mast seed first arrives, despite density being low at the start of the mast year. In having found a bait, acceptance of a meal is a function of familiarity with the bait. This is important for rodents. They're encountering a completely novel food, and they don't like to take big meals of novel foods. This is why we pre-feed, but we only pre-feed once, and pre-feed doesn't visibly last long on a forest floor with lots of rats in it. Bait acceptance also likely influenced by how hungry the animal is and how appealing the food seems, for obvious reasons. And the ability to move baits around and consume in safety might also be important. Now, the way this big study of mine worked wasn't to start with this framework and then devise good measures of all these things so we could tease out exactly where we're going right and wrong. What's happened is that we've scrambled together a data set from other people's 1080 operations, many of which we didn't even visit, but where they were doing tracking tunnels, and into some of which we intentionally introduced experimental blocks. And then we modelled all these variables, all the variables we could actually score or could find a good proxy for, and then viewing the results within my little framework. For example, everyone wants to know if our best practice sowing rates are right. So a modelled sowing rate in pellets per hectare. So if there's not enough bait, some animals will miss out because no bait's landed in their home range, or there wasn't enough bait to overcome all the bait consumers out there. So a modelled sowing rate per se seems kind of simple, but these ecosystems are often masting ecosystems in which rodent populations go up and down a lot. in response, and ranging behaviour probably changes too. So modelling sowing rate per se isn't that helpful in the context of these dynamics due to all the noise that masting generates in bait competition and ranging behaviour and density. Ideally, we would model sowing rate in interaction with bait competition and ranging behaviour to allow sowing rate to make a difference only when there's heaps of rats or when home ranges are smaller and there's lots of background food. In reality, we modelled proxies for bait competition and ranging behaviour. Our proxy for bait competition was rat density. We didn't have any measures of possums or deer at most of these sites, and the other two, which are the other two primary bait consumers. Our proxy for ranging behaviour was the population growth rate, which we surmised should reflect background food and therefore also home range size. We also tried this with delta T for a beech forest only version of the analysis, but my main analysis included all forest types. If a sowing rate interaction with rat density was supported, that would signal that bait competition explains our poor results, and we just need to sow more bait when there's many rats. If a sowing rate interaction with background food matters, that's signalling to me a ranging behaviour issue, which we could address by sowing more bait, or perhaps by sowing it more precisely, or maybe more easily by waiting for background food levels to drop and home ranges to consequently expand. Now, as we've heard a lot about, there was a massive beech mast in 2019. Beeches and Rimu and tussocks actually all masted at once. Despite our best practice sowing rates, which we'd spent quite a lot of angst over agreeing on in our tag meetings, the first operation in Kahurangi in 2019, in the presence of abundant background food, beech seed, didn't go very well. The operation was called COB 2019. Rats were increasing and at moderately high tracking rates at the time of the operation in June. About 17% of the rats survived, and numbers consequently rebuilt to damaging levels quite quickly. An emergency tag meeting was called, and despite the sowing of some 250 pellets per hectare, it was way off Dean's chart of rat densities versus tracking tunnels. I think we went up to about 12 rats per hectare. Some tag members still suspected that we had a bait encounter problem rather than a bait acceptance one. So we devised a strategy to test this once and for all, which was to conduct a series of highly over-spec 1080 drops during the rest of the mast year and then into the post-mast year. The over-spec 1080 drops look like this one on the right. Cob is on the left, with 250 pellets per hectare sown with very little overlap of swaths. The green lines there are the helicopter flight lines, which were 180 metres apart based on the calibration of the buckets, which demonstrated reasonably reliable spread out to 90 metres each side of the flight line. For the following 7 1080 drops during that mast year, we halved the flight path spacings, creating total swath overlaps and increased the net sowing rate to 333 pellets per hectare. We didn't get any extra money for all this, extra helicopter time and bait. It came at the cost of huge reductions in the area of forest treated that year. But we agreed that if this didn't work, we'd give up on bait encounters and focus on bait acceptance. And this is how it went. This chart again has a bar for each operation showing the percentage of rats that survived according to tracking tunnels. I've squeezed it all down into the left because I firmly believe that all charts of tracking rates should have a y-axis up to 100%, but I'm going to break my rules so you can see it a bit better and zoom into the bottom bit there. You can see the cob operation over on the left that's there with disappointingly high survival rate, and then each of these blue bars is an overspec operation with those super tight flight path spacings, and that little 100 is shouldn't be there, so just ignore that. And you can see along here there's some operations that are shown as just a little dash. Hopefully you can see those, which are operations that got 0 afterwards. So it doesn't look like it here, but modelled out with all the other variables and all the other mast year operations across the other mast years, this overspecking is actually supported as a slight improvement attributable to the overspecking in rat survival or rat kill rates. But the 3% survival target was generally not met, and rats recovered quickly in most of these sites. There was no effect of rat density or rat tracking rate itself, neither by itself nor interaction with the increased sowing specs. That was operations with low to moderate rat density still got disappointing results during the period of high background food levels. Then after the seed had germinated in the late spring of 2019, kill rates of rats improved a lot, despite the expected increase in trackability during this period. As Dean was saying, we expect trackability, and I think Joe perhaps to increase during this period. So your chances of getting a zero, it's going the way it's going indicates that actually these kill rates were really quite good. For mice, the overspec operations also, when modelled out across all of the data set, did generate a significant improvement in mouse kills. But again, mouse survival rates horrendously high. But also, again, a massive improvement in mouse kill rates after that seed had germinated. Still, not quite so good as for the rats, but still some very good rat kills, mouse kills coming through in the post-mast year. So turning back to the framework, we'd ruled out field life problems by modelling rainfall after baiting and bait competition, at least from rats. So what this tells me was that home ranges very likely get smaller in the presence of background food, and thus expensive baiting helps a bit. But the persistence of the problem, despite the overspec baiting, and then the vanishing of the problem after germination, told us we had issues with bait acceptance in the presence of high background food levels. So for rats, this leaves a combination of insufficient familiarity with the novel food, low hunger levels, and perhaps poor relative palatability of baits compared to beech and remu seeds as the cause of the problem. Now, as I said, we'd pre-fed all the operations once to overcome the familiarity hurdle. Perhaps in the presence of high background food, one pre-feed isn't enough. Perhaps the rats would love to eat the bait, but because they're just not hungry, they can afford the wisdom of applying a more rigorous testing regime, taking a little longer to get convinced that it's safe. We could test this by increasing pre-feed sowing rates, or perhaps by doing two pre-feed applications. We've done experimental manipulations of pre-feed sowing rate, and it didn't help, neither in the presence nor absence of mast seed. We simply can't really afford two pre-feed applications, so there's no real appetite to test this. Alternatively, the rats may be perfectly familiarised with the novel food, but like humans and other omnivores, they prefer a mixed diet when they can get one, so their meals are made up part by seed. No matter how palatable the bait or how familiar with it they are, they simply don't get a lethal dose in one mixed meal of seed plus bait. It's really tricky to test this kind of thing in the field, and also quite difficult to solve other than by waiting for background food levels to decline. Which we did, and in the last operations in the post-mast year, kill rates improved massively, despite actually an eventual return to lower sowing rates and looser flight path spacings once we'd seen that starting to manifest. How am I going for time? I can't see my clock either.
You've got 6 minutes, Josh.
6 minutes. Okay, cool. So exactly which of the acceptance factors is operating, we can't really tell, but maybe we don't really need to know. All right, 6 minutes on stoats, and me and Susan actually grabbed the same photo off the internet, didn't we? Here we go, that was a fluke. Stoats and feral cats when the bait is the rats and mice rather than the cereal pellet, because it's all secondary poisoning. So with secondary poisoning, I can't see any issues on the encounter side of this framework. Stoats have large home ranges, and they probably adjust them a bit to match rodent abundance, so they shouldn't have any trouble encountering rodents even when density is reasonably low. And I've added a fifth thing to the acceptance side, which is catchability. So they're already, on the acceptance side, they're already familiar with the bait. They can easily carry rats and mice around, so I've crossed out another couple of those. But when we poison some rodents, but not others, and usually, as I've shown, we've left behind live mice, sometimes in large numbers, when a stoat encounters a poisoned rat or mouse, it may not be very hungry, so it might only eat a small bit and get crook but not die. Perhaps a poisoned prey also is less palatable. We know stoats and cats will scavenge dead prey, but given a choice, they might prefer freshly killed mouse over a potentially risky sick or dead rat or mouse. So thinking about all this, and I know Susan said that that the tracking tunnel data is suggesting that that aerial 1080 secondary poisoning of stoats is generally pretty good. I'm a bit worried about it, partly because the sites through the program in which we've struggled to actually recover birds properly are the sites where we've tended to have lots of mice and tended to not poison them very well. So I've been asking myself the question, have unpoisoned mice been messing with our stoat control? Got this little sort of series of graphics that kind of demonstrate what I think happens. We've got live rats, live mice, dead rats, and dead mice. Hopefully you can distinguish them through. So I think in a situation where we've started off with a lot of rats and we've killed them all, giving this and no mice in the system, we've demonstrated pretty clearly, and this is Elaine Murphy's study in Puriura was like this, that stoat secondary poisoning is quite effective. It's probably still quite effective if there's supposed to be a whole lot of poison mice and rats in that one. That we do pretty well too. What happens if we only poison half the rats? Actually, looking through, eyeballing the data set, as I said before, I think it's starting to not be quite so complete. And then if we poison the rats and only half the mice, or as often we do, we poison the rats and none of the mice, that we might be doing the equivalent of mixing pre-feed pellets with toxic pellets when we sow them. Looking through the operations that we've got and trying to put them on a kind of axis here where we've got the ratio of toxic to non-toxic rodents, and with forgetting about whether they're rats or mice, and then the density. So I've got this sort of up here, we've got lots of toxic rodents, very few non-toxic ones, versus a low density rodent population where we've killed most of them, or a high density rodent population where we've left most of them alive, for example, as we do in these eastern valleys with low rat numbers and high mouse numbers, or a low rodent density population where we've left most of them alive. I've gone through and put a whole lot of operations, looked at case studies for a whole lot of these operations. I'm pretty convinced from looking at these, and as I said, I haven't crunched these numbers properly, that we're okay when we're in this corner. I think we're actually okay when we're down in this corner too. But when we start getting across to the other side, the data set's actually not really able to tell us how we're going. And that's because a lot of these eastern valleys where we get high mouse numbers and don't kill many of them, there's all this confounding between stoat trapping and tracking tunnels. In this case, this is the South Huranui where we've got a 1080 drop shown in pink shading, and the blue lines are stoat traps, and the little dots are stoat tracking tunnels. The other problem is that our survey program often doesn't give us before or after, and partly that's because these 1080 drops happening in late spring happen across that period when the new cohort of young stoats arrive. So it doesn't really work very well for the before or after treatment, non-treatment framework. And those kind of problems are apparent, in the Dart, Capels, the Eglinton. So it's really difficult to get a handle on whether or not mice are actually messing with our stoat kills by looking at tracking tunnel data. But I figure that if background food matters for rats and mice, then it could well matter for stoats, and we should be asking ourselves that question. That's me, thank you.
Thank you, Josh. That was perfect timing.